So, Prashant sir,
Yes, sir. Yes, I will do.
make a post and also all the speakers.
Julie, madam, you can go.
Co-host,
You can share now.
good morning, Dr. David Paul. Good morning.
Good morning, sir. Good morning, sir. Good morning, Chandradas, sir.
Good morning. Good morning.
Good morning, sir.
David is working in Naipur, Calcutta.
Oh, okay.
He's an assistant professor.
Sir, is it visible?
Not it. You can double click, no? It's coming. Madam join ചെയ്താ Okay, Julia, PowerPoints ഒക്കെ ready ആണല്ലോ ഇല്ലേ? Okay, now it's okay. Keep like this, better.
Sir,
Yes, sir.
the live stream started now, sir.
Okay, thank you. Hello. Hello, sir. Ah. Good morning. Julie Madam, shall we start now? Now it is 10 o'clock, 11 o'clock. Julie Madam? Julie Madam, shall we start now? Good morning, Dr. Julie. Dr. Julie, shall we start now?
Yes, sir.
Right, yes, now time 11 o'clock.
So, is it visible, sir? It's okay?
Visible, yeah.
Okay. Respected dignitaries, honorable speakers, esteemed colleagues, researchers, dear students, and all participants joining us through Zoom and YouTube. A very warm welcome to this special occasion of National Science Day. It is organized by Kerala State Branch of Indian Pharmaceutical Association. Myself, Dr. Julie Gurian, Faculty of Pharmaceutics, Lisi College of Pharmacy, Ernakulam. With humility and gratitude, I take up the role of Master of Ceremonies for today's National Science Day 2026 program. Science is not just about discoveries. It is about curiosity, courage, and above all, the courage to question. Today, we gather to celebrate a day that transformed the course of Indian science. Every year on February 28th, India celebrates National Science Day to commemorate the historic discovery of Raman Effect by Sir C.V. Raman in 1928. This remarkable discovery is far beyond the confines of laboratories. It earned India its first Nobel Prize in Science. Indeed, it was a defining moment, a true turning point in India's scientific journey. Being a woman academician, it's a matter of pride for me to share the theme of National Science Day 2026. Women in Science, Catalyzing Vikasit Bharat. This theme is both timely and transformative. Today, women scientists, researchers, educators, and innovators are breaking barriers and leading innovations in healthcare, pharmaceuticals, biotechnology, space science, and artificial intelligence, fields that are shaping the future of our nation and the world. As India moves towards the vision of Vikasit Bharat, empowering women in science is not a choice, but a necessity for inclusive growth, scientific progress, and a sustainable national development. Today's program reflects this vision by bringing together eminent scientists, industry leaders, academicians, and young researchers, many of whom represent the growing strength and leadership of women in science and pharmaceutical research. May this celebration inspire our young minds to dream big, innovate boldly, and believe that science knows no gender, only potential. Let us now formally commence the proceedings of National Science Day 2026.
Vande सुजलां सुफलां मलयज शीतलां सस्यश्यामलां मातरं वन्दे मातरम् शुभ्रज्योत्स्ना पुलकितयामिनीं फुल्लकुसुमित द्रुमदलशोभिनीं सुहासिनीं सुमधुरभाषिणीं सुखदां वरदां मातरं वन्दे मातरम् कोटि कोटि कंठ कल कल निनाद कराले कोटि कोटि भुज द्रुत खर करवाले अबला के नुमा ए तो बले बहुबल धारिणीं नमामि तारिणीं त्रिपुदलवारिणीं मातरं वन्दे मातरम् तुमि विद्या तुमि धर्म तुमि हृदि तुमि मर्म त्वं हि प्राणा शरीरे बाहुते तुमि मा शक्ति हृदये तुमि मा भक्ति तो मारई प्रतिमा घड़ी मंदिरे मंदिरे मातरं वन्दे मातरम् त्वं हि दुर्गा दश प्रहरणधारिणी कमला कमलदलविहारिणी वाणी विद्या दायिनी नमामि त्वां नमामि कमला अमलां अतुलां सुजलां सुफलां मातरं वन्दे मातरम् श्यामलां सरलां सुस्मितां भूषितां धरणी भरणी मातरं वन्दे मातरम् वन्दे मातरम् वन्दे मातरम्
Thank you all. Please be seated. Now, I respectfully request all dignitaries and participants to please be seated, and we shall now begin with the welcome address. I invite Dr. Anjana John, Principal, JDT Islam College of Pharmacy, Kozhikode, to address the gathering.
Good
Over
morning.
Good morning, ma'am.
Good morning. Good morning, all. Distinguished guests, eminent scientists, revered academicians, and dear students. On this day of commemorating the discovery of Raman Effect, let us celebrate everyday scientific wonders and foster innovation and research for sustainability. On this occasion, I extend a warm welcome to Dr. Jayashekhar, President, Kerala State Branch, to preside over this function. IPA Kerala State Branch is now the best state branch, winning multiple awards under the visionary leadership of Dr. Jayashekhar. On behalf of IPA Kerala State Branch, I extend a warm welcome to our chief guest, Professor Chandrabas Narayana, an eminent scientist and professor at Jawaharlal Nehru Center of Advanced Research, Bangalore. We also have with us Dr. Sriraj Gobi, Chairman and Managing Director, Molecules Group, Thrissur, as guest of honor. A warm welcome to you, sir. I extend a hearty welcome to Dr. Sabita M., Principal, Amrita School of Pharmacy, and Dr. Arun Rashid, Vice Principal, Al Shifa College of Pharmacy. A warm welcome to Dr. David Paul, Assistant Professor at NIPER, Kolkata. IPA Kerala State Branch extends a warm welcome to the young scientist Dr. Ashwati R. Devan, DST Inspire Fellow at Amrita School of Pharmacy, and Dr. Dylan P.N., Associate Professor at Nupens College of Pharmacy. A hearty welcome to Dr. Bobby Jones, Professor of Pharmaceutics at St. Joseph's College of Pharmacy. A warm welcome to all IPA members, faculty, and dear students. May this National Science Day instill scientific temper and inspire faculty and students. Thank you.
Thank you, ma'am, for your inspiring welcome address. We shall now have the presidential address. It's my honor to invite Dr. P. Jayashekhar, President, Indian Pharmaceutical Association Kerala State Branch, and former Dean, College of Pharmacy, National University of Science and Technology, Muscat, to address the gathering. Over to Jayashekhar, sir. Sir,
Jayashekhar sir, you are muted.
Yeah, good morning to all. Good morning to all.
good morning, sir. We can hear you.
Good morning to all. Thank you, Julie. Chief guest of the day, Dr. Chandrabas Narayan, Professor of Jawaharlal Nehru Center for Advanced Scientific Research and former director of Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. And he's well known for his studies in Raman Effect as to Raman spectroscopy. And we are truly honored to have him here in this National Day. And eminent scientist as well as entrepreneur, Dr. Sriraj Gobi, Chairman, Managing Director of Molecules Group, Thrissur, invited young scientist Dr. Ashwati Devan and Dr. Dhilan, respected dignitaries, fellow pharmacy teachers, dear students. Thank you very much for joining this initiative of IPA and on the National Day of National Science Day. It's indeed a proud moment for all Indians to celebrate National Science Day on 28th of February. And this day commemorate the discovery, as we know, this day commemorate the discovery of Raman Effect by C.V. Raman, which brought the first Nobel Prize in Science to our nation in 1930. We started observing National Science Day from 1988. And this occasion reminds us the immense power of science to transforming society and improve human life. India is blessed with highly talented scientists and technologists, engineers and educators. Yet the infrastructure in many of our education institutions, research centers, industry is still not adequate to fully accelerate cutting-edge research and innovations. The percentage of GDP spent on science and technology in India is declined from 1.6% in 1960 to 0.6% in 2025. In contrast, countries like Israel spend 2.4% of GDP, United States 3.5, China 2.4 of their GDP on R&D. For sustainable growth of science and technology, India must aim to increase the investment at least 3% of our GDP in R&D. This called for a collective commitment from both the government and industry to strengthen funding, modernization of infrastructure, and promote vibrant, innovative ecosystem in the nation. India has to work together with the urgency and vision to build a strong, self-reliant, innovative, driven nation. As in the introductory remarks, Dr. Julie mentioned the theme of this year is Women in Science, Catalyzing Vikasit Bharat, highlights the crucial role of women playing in building, developing self-reliant India. For a long time, women faced many barriers in education and scientific careers. Yet, despite of these challenges, they have made remarkable contribution in every field, from space research to medicine to engineering to technology. Translating these achievements into national progress for Vikasit Bharat is the need of the hour, and we have to work collectively for this mission. India has produced many inspiring women scientists, such as Kalpana Chawla, who symbolizes courage and determination, an astronaut and astrophysicist. Tessie Thomas, a popular scientist in known as a missile woman of India. Asmi Chatterjee, who is a medicinal chemist, worked on anti-malarials and epilepsy drugs. Janaki Ammal, a renowned plant scientist and cytogenetics. Yamuna Krishna, a well-known scientist on DNA nanotechnology. Kiran Mujumdar Shah, a founder of Biocon, who worked on biotechnology, transformed biotechnology into pharmaceutical products for making affordable life-saving medicines. Women in science is not just contribution, they are catalysts of our progress. Dear students, remember that curiosity, hard work, confidence can take you anywhere. Science belongs to everyone, and our idea can shape our future. The future of medicine and healthcare lie on your hands, and your research and innovation can serve countless members. Science education research must be always a purpose and not merely for a degree. As a teachers, we have the moral responsibility to nurture the curiosity, inquisitiveness, critical thinking, and problem-solving ability of the next generation. Let me conclude my words with the famous quote of C.V. Raman: "Ask the right questions, the nature will open doors to her seekers." Let us pledge today to support and promote women in science, so that together we can build a strong, innovative, and developed India. Thank you. Happy National Day. Jai Hind.
Thank you, sir, for your inspiring and visionary address. A special highlight of today's celebration is the National Science Day message on the theme Women in Science, Catalyzing Vikasit Bharat. This important message will be delivered by Dr. Sabita M., Principal, Amrita School of Pharmacy, Ernakulam, a leader who exemplifies how a woman in academia inspires generations of scientists. May I now invite Dr. Sabita M. to kindly deliver the National Science Day message. Over to Sabita ma'am.
Thank you, Dr. Julie, for the kind introduction. Hope I am audible.
Yes, ma'am.
Yeah. Good morning, respected dignitaries, dear colleagues, and my dear students. It is an honor to speak on the theme Women in Science, Catalyzing Vikasit Bharat, the theme for this year's National Science Day celebration. Dr. Jayashekhar already gave a very good overview, but as this is the topic given to me, let me try to speak for a couple of minutes. The vision of Vikasit Bharat, a developed, self-reliant, innovation-driven India, is not merely an economic aspiration. It is a scientific, technological, and social transformation. And this cannot be realized without fully harnessing the intellectual power of women also in science. From agriculture and defense to pharmaceuticals and public health, as Dr. Jayashekhar mentioned the examples, we know women scientists are not only contributing but are transforming the nation's growth trajectory. India's scientific history offers powerful examples. He already mentioned a few. In agriculture, as he mentioned, Dr. Janaki Ammal made pioneering contributions to plant cytogenetics and crop improvement, strengthening agricultural productivity and food security. In defense science, as we all know, Dr. Tessie Thomas from Kerala, known as the Missile Woman of India, played a key leadership role in Agni missile program, reinforcing India's technological self-reliance and strategic strength. In space research, we have the example of Dr. Ritu Karidhal, contributed significantly to India's Mars orbiter mission, demonstrating India's scientific capability on a global platform. Equally transformative are women in the pharmaceutical and health sectors. Couple of examples are Dr. Indira Hinduja, who pioneered the assisted reproductive technologies in India, delivering the country's first test tube baby and advancing clinical reproductive medicine. Dr. Gagandeep Kang has led impactful research in enteric infections and vaccine development, strengthening India's public health research ecosystem. Kiran Mazumdar Shaw, through Biocon, transformed India's biopharmaceutical landscape by making affordable biologics and insulin accessible, positioning India as a global leader in biosimilars. So these women exemplify how science, when combined with leadership and perseverance, catalyzes national development. However, while celebrating achievements, we must acknowledge the challenges that women continue to face. Professionally, women often encounter underrepresentation in leadership roles, limited access to research funding, unconscious bias, and fewer opportunities in high visibility projects. The leaky pipeline in science, technology, engineering, and mathematics remains a reality. Reasons may be different, but then it is a reality, with many talented women leaving research careers midway. This we usually see in colleges also. As a girl student, very enthusiastic and performing very well, may not be able to continue. Many of maybe because of the family background and the reasons from the family. On the personal front, women frequently balance demanding scientific careers with family responsibilities, caregiving roles, and societal expectations. Career breaks for maternity or caregiving can affect research continuity, publication output, and promotions. The struggle is not about capability, but about navigating structural and social barriers. In today's program, we have our own PhD student, Dr. Ashwati, giving a session. I know how difficult it was for her to manage her baby and then pursue her PhD. So usually women face these challenges in their personal and professional lives too. If India aspires to become Vikasit Bharat, we must move from symbolic inclusion to systematic support, mentorship, flexible research pathways, equitable funding policies, leadership training, and institutional cultures that value diversity. A developed nation is built not only through infrastructure and technology, but through inclusive opportunity. When women scientists thrive, innovation accelerates, healthcare will improve, and society will progress. I'm proud to say that at Amrita, we have many women in leadership roles. Our chancellor herself is an example how a woman can lead. Women in science are not merely participants in India's development story, they are catalysts of transformation, as I said earlier. So let us commit to enabling their journey, strengthening their voices, and ensuring that the path to Vikasit Bharat is truly inclusive. I take the opportunity to thank Dr. Jayashekhar and other leaders of IPA Kerala State Branch for organizing this program in connection to National Science Day. We are honored to have renowned scientists like Dr. Chandrabas Narayan and an esteemed entrepreneur, Dr. Suraj Gopi. I thank them for taking their time out and joining us for this program. I wish all the scientists, all students, and all my colleagues a very pleasant day and hope that we all will try to contribute our level best to Vikasit Bharat. Thank you. Thank you all.
Thank you, ma'am. Your words have powerfully highlighted the vital role of powerful Indian women like Kalpana Chawla, Hinduja in science in shaping Vikasit Bharat. And moreover, you could pinpoint the challenges the women of today face in field of science. We can all hope that these challenges can be overcome in the near future. Thank you, ma'am. Now, we are deeply honored to have with us our chief guest and keynote speaker, Professor Chandrabas Narayana. He is the professor of Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, and he was the former director of Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram. Now I invite Dr. Arun Rashid, Vice Principal, Al Shifa College of Pharmacy, Perindalmanna, to kindly introduce the chief guest. Over to Arun Rashid, sir.
Thank you, Dr. Julie. Is it audible, madam?
Yes, sir.
So, good morning, one and all. Dr. Jayshekhar, sir, IPA President, Kerala State Branch, distinguished dignitaries, faculty members, and dear students and esteemed guests. It's a matter of immense pride and privilege to introduce our chief guest for the National Science Day 2026 celebrations organized by Indian Pharmaceutical Association, Kerala State Branch. We are deeply honored to welcome Professor Chandrabas Narayana, an eminent scientist, distinguished academician, and visionary research leader, currently serving as a professor at Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, and former director of Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, Kerala. Professor Narayana completed his doctoral studies at prestigious Indian Institute of Science, Bangalore, and subsequently pursued his postdoctoral research at Cornell University. Since joining Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore in 1998, he was made a remarkable contribution to interdisciplinary science, establishing himself as a global authority in Raman spectroscopy and material research. A trained condensed matter physicist with an extraordinary interdisciplinary vision, Professor Narayana has pioneered the application of Raman spectroscopy in diverse dimensions, including drug-protein interactions, pharmaceutical screening, protein structure function analysis, and biomedical diagnosis. His research seamlessly integrates physics, chemistry, and biology, exemplifying the spirit of modern scientific innovation. During his tenure as director of Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, he played a transformative role in strengthening biotechnology research, fostering collaborative scientific ecosystem. His academic excellence is reflected in more than 180 international publications, influential patents nearing commercialization, and numerous prestigious honors, including the Materials Research Society of India Medal, Sir C.V. Raman Young Scientist Award, and the Platinum Jubilee Lecture Award of Indian Science Congress. A fellow of various prestigious academics like National Academy of Science India, Royal Society of Chemistry USA, and Indian Academy of Science, Dr. Narayana contributes to inspire the scientific community through the leadership, scholarship, and commitment to interdisciplinary research. Today, we have privilege to have him inaugurate this celebration and deliver a keynote address on Raman spectroscopy, basics and use in drug discovery, a interdisciplinary approach, a topic that beautifully reflects the convergence of fundamental science and pharmaceutical innovation. On behalf of Indian Pharmaceutical Association, Kerala State Branch, and all gathered here, I extend warm and respectful welcome to Professor Chandrabas Narayanan and invite him to inaugurate and program and address the audience. Thank you very much.
With great respect, I now request Sir Chandrabas Narayana, sir, to enlighten us with inaugural and keynote address. Over to sir.
Can I share?
Yes, sir. Yes, sir.
Yeah. Yeah. Good morning. I think I have 30 minutes, so I'll try to finish my talk in 30 minutes. But first of all, let me thank Dr. Jay Shekhar for inviting me to be the speaker and chief guest for this lovely program, which is organized by the Indian Pharmaceutical Association, Kerala State branch. And I was pleasantly happy to hear that this is one of the best chapters which is working and receiving accolades from the national level. I would like to thank all the dignitaries who are attending this, the students, and also the staff. So with this lecture, I'm formally inaugurating this National Science Day program. And let me tell you that I work, as was mentioned by Dr. Rashid, that Arun Rashid, that I work with Raman spectroscopy. So I take a pride in actually propagating what Raman actually discovered. And this is very important today because Raman has actually multiple roles played in science, and everyone, young and old, should know why this Raman spectroscopy is such a wonderful discovery, and this should motivate us to come out with many different discoveries. Today, I am actually going to focus on telling you about Raman spectroscopy, and then I would like to actually show you with one example only, because of the lack of time, how we can actually use Raman spectroscopy in drug discovery. It's a completely interdisciplinary approach, and I promise you that I do not need more than your 12th standard knowledge to actually talk to you about this particular talk. So basically, when we are talking about Raman spectroscopy, we are talking about light interactions with material. And this light interaction with material has multiple things which you all know. Initially, the light would actually get transmitted or reflected, depending on what is the opacity of the material. But also it will have absorption, which is a very important function. Alongside with this, it also gives out luminescence. And this luminescence is a very important aspect because you know that you have something called fluorescence and phosphorescence associated with it. This is actually another interesting property which comes in. But alongside with this, I would like to say that there is a very interesting and very important phenomena which happens, and that is elastic scattering. And when I say elastic scattering, all of you remember something called elastic collision. And elastic collision is nothing but basically when a ball goes and hits a wall, it comes back with the same energy it went in. So there is no change in energy. That is called elastic. And similarly, in the case of light, when it is scattered elastically, the color of the light will not change because energy is related to the frequency, and energies, the frequencies are having different colors. So there won't be any change in frequency, hence it will be the same color. But what Raman actually discovered is it actually said there were inelastic scattering, just like inelastic collision, where if I throw a chapati dough onto you, it will not come back with the same energy, it may stick to you. So there is a change in energy which happens, and that means the change in color in the case of light. And that can be red shifted or blue shifted. Here I have shown it as red and blue. It doesn't mean that it is exactly always red and blue whites. Green can shift to slightly towards the red side of the green, or green can shift to the blue side of the green. And that is what I mean by red shifted and blue shifted light. So if I want to actually show you that this indeed happens, then I would be able to do this experiment in front of you. And that's a very simple experiment. Even you can actually try to do that. But at this point of time, I wanted to actually ask you a question that which you can try to answer at some other point of time. How do I see you? Or how do we see each other? Which of these phenomena are reason for us to see each other? So that I leave it as a experiment or a question to answer by all of you. Now, when we talk about materials, we can say that Raman can look at anything from originating from physics, chemistry, biology, geology, and beyond. So Raman actually encompasses all the fields. And that is why this is one of the greatest discovery in the area of physics. And this Nobel Prize was very, very well deserving. Now, I am showing you here a laser light, which is a monochromatic laser light, which is a blue color light. And you can see in the dark room, you can see the dust particles scattering. And this is the elastic scattering coming from the dust particle. Same time, this light is passing through this liquid, which is transparent. And you can see this liquid is nothing but liquid cyclohexane. And you know cyclohexane is a transparent material. And now my camera is 90 degrees to the laser. And all of us know the laser travels straight line. So if I am seeing 90 degrees to the laser, that means it is the scattered light coming out of the liquid. And now you don't get confused. This is not white. It is because the light is so intense that it actually makes our detector or the camera saturated. And that's why it's looking white. You can even see at the starting point of this laser also it looks white because it is too intense at that point of time. Now, if I want to show you that there is a shifted frequency, what I have to do is I have to actually filter out this blue light. And nowadays, you know, very good filters exist. And these filters can completely remove a particular wavelength of light. Here you can see I have put the filter and you can see the disappearance of the dust particles scattering. The blue light is not coming. But all of a sudden you see that indeed your liquid is giving different color. And this is what is the Raman scattering coming out of this liquid. And this is what got the Nobel Prize for it. Now, how do I know what light is coming out of this? For this, you know that whenever you have multiple lights coming out, you can use a prism or a grating which can disperse the light. And you know that white light, when goes through the prism, becomes 7 colors. So same way, you can use prism. So this knowledge was there with Raman. And Raman actually went ahead and did this experiment using this particular spectrometer, which is actually present in Indian Association for Cultivation of Science in Calcutta. That is the first Raman spectrometer which was used. And he used to be happy to show people how the Raman effect happens. So here you collect the light, you pass it through your sample, and then you collimate the light and make it to fall on a prism. And the prism actually disperses the light, and you have a photographic plate which actually records the spectrum. And the first Raman recording is shown here. And you can see the intense light which is there in the center is the elastic scattered light. And this is the red shifted light. And this is the blue shifted light. And this got the Nobel Prize for Raman from an organic molecule. Now, today, you can make very simple Raman spectrometers where you can take a laser which is about 1,000 rupees, and you can actually, this is a green laser, and you can make it monochromatic by putting a filter and then pass it through your liquid or any material and then filter out the green light, and then you collect it using a fiber, all the light scattered in this direction, and then you take that fiber and put it onto a mirror and put it on top of a grating instead of a prism, because grating is a better dispersing agent. And now it disperses the light, and you can use a camera which is similar to a SLR camera and take a photograph. And here I am showing only the red shifted light. you can see that this is the elastic scattered light, and then you have different lines which are coming out, and it goes all the way till red, right? So now you can realize that if I digitize this lights, I can get spectra out of this. Now, this is what is called the Raman spectra. And the interest is not in the laser light or where the light has come out, but interest is in the how much did this light shift. So this energy is related to vibrations of the molecules or atoms in a solid. So this is what is very important, because depending on the environment in which they are present, they would actually have a specific interaction with the light and produce these lights. So when I take any two atoms together, any two atoms, hydrogen, H2, O2, N2, or any of those, will have only one vibration associated with it, which is symmetric stretching. And here I have taken HF and HCl. You know that the fluorine is lighter than HCl. So you can see that you have this vigorously oscillating, while this is very sluggishly oscillating. As I always say, is whenever things are lighter, they are more energetic compared to the heavier atom. So this is what is happening. So I have given a unit of centimeter inverse, but you can realize centimeter inverse can be converted into an energy term by multiplying with Planck's constant and velocity of light. And simple conversion is 8,000 centimeter inverse is 1 electron volt. And to get in perspective, 1 electron volt is 1.1 electron volts is the band gap of a silicon. Now, you can realize this is about 500 milli electron volts, and this is actually 300 milli electron volts. So that is the energies at which these vibrations happen. So the shifts would be around that region for this if we do the Raman. Now, when I actually look at water molecule, which is actually looks like this is the oxygen and the two hydrogen. There are three vibrations associated with it. This is symmetric stretching, this is asymmetric stretching, and this is symmetric bending. Now you can realize bending a bond is easier compared to the stretching of the bond. And that's why the energy of the bending is always less than stretching. Now, any atom which looks like this will have this kind of a vibration. And one example you all know is CH2. CH2 is actually exactly looking like water molecule. And now you can realize the CH2 would actually oscillate exactly like water molecule. This is symmetric stretching, asymmetric stretching, and symmetric bending. But CH2 is very unique in the sense it cannot exist independent like water molecule. It actually has an R group associated with it. Now, when this R group is there, it is like a heavy thing holding the CH2. So when you hold this CH2 at this point, it can also do these motions. Like this is called twisting, the two atoms going in and out of the plane of the paper. And this is called rocking, which is like twisting in direct in the plane. And you can also have wagging, just like the dog's tail wagging, both the atoms going in and out of the system. So suddenly you can see, just because of this extra R group, these vibrations are different. So actually these vibrations and their energies are so unique that they become fingerprint of the molecule. And people have been using this as a characterizing tool. So what happens is that you can actually characterize any of these materials. And today we make, this is a spectrometer made by me for portability to take it to point of care. So you have a microscope objective which is present. Your sample can sit here. You take a fiber, put it onto a spectrograph and connect it to a laptop and take a recording. This is the zoomed in version of it. For diagnostics, which I do quite a lot, I can use a 96 well plate and move it under this microscope, and I can actually do a pathology lab experiment. So which is what another area which I actually specialize in. Today, Raman spectrometers look similar to a mobile. So you can see a glass slide can be put inside, the laser is inside, the spectrograph is inside, and you can get the spectra on top of it. So it's very easy to do these things portably. Today, these are available. So one very interesting thing is you buy organic fruits, and the idea is the organic fruits do not have pesticides, but you pay a lot of money. If I want to be sure that there is no pesticide, I can just put in the laser light and record the spectra, and I can actually get to know whether there is a pesticide in this system. So that simple it is. It can be used in areas to find out these kinds of things. Now, today in biology, there is cancer detection is becoming very important. And you know that for women, one of the biggest problem is breast cancer. And in the case of breast cancer, you are supposed to take a mammogram. Now you can realize if it is a fatty breast, then the lesion which is there can be easily detected. But if the breast happens to be dense, then you can't see a lesion very easily in mammogram. So this is something which makes it harder for mammograms to detect it. Now this is where Raman can come in. Another area where the Raman actually helps is when you want to remove a tumor, you want to remove all the cancerous tissues away. So there is a reason why we need to look at something called your margins. You have to look at. So here you can see that this is a 1 second spectra of the tissue while the surgery is happening. And the statistics is shown by the red line. And you can see that this is a normal tissues, this thing. So you know that doctor knows this is a normal tissue. And if you look at the malignant tissue, the spectra is quite different. Again, the red is the statistics of that, of the same patient. And you can see this is like a light and day. So this can guide the surgeon to remove all the cancerous tissues without leaving any tissue back in the patient. And this is very important. And that's why you don't need to go for mastectomy. Instead, we can remove the tumors completely. So this is happening today. In addition to this, because Raman is sensitive to various different biological systems, you can use for histopathology. Here you can see the histopathology shows 2 epithelial tissues of a urinary bladder. And you can see this much more distinctly in Raman without staining. So this is the advantage. And today this staining, this is a pseudo color. And you can see that you can actually do a histopathology using Raman. And it can be up to a micron level accuracy. So another area where most of the pharmaceutical companies would be interested is today we want to actually do a QC or you want to find out fake drugs. So here is an example of a paracetamol. And all of us know that paracetamol doesn't have only paracetamol. It has aspirin, it has caffeine, and it has cellulose as a binder. Now, the paracetamol is in a particular ratio, in a particular this thing. Suppose let us say it is Dolo 650 or it is Calpol or any of those things. There will be a ratio which will be very uniquely done for a system. Now, I can do a imaging of this without taking it out of the capsule. And then if you zoom in, you can start seeing all the components. So the blue is the caffeine. Green is the actually paracetamol, and orange is the actual aspirin, and yellow is the cellulose. And you can see, you can zoom it up and see. So the QC can be done or fake drug detection can be done. And today, more or less, many of the pharma companies are actually having Raman spectrometers in their place. But normally, we are not always happy using Raman directly. Many a times with therapeutically important proteins, we may need to have an enhancement factor. Here is where we actually take nanoparticles of silver, which is very easy to make. You reduce silver nitrate by sodium citrate and you produce these silver nanoparticles. And they beautiful nanoparticles are formed. And these nanoparticles, when it's incubated with your protein in this ratio, you actually do a incubation and then you do a centrifuge to make droplets on a hydrophobic surface. So now you have protein which is incubated with the silver like this. And then when it is put under this, we actually can get very strong signal from a sample. Now, this is an example of that called surface enhanced Raman, which is nanoparticle attached. You can see when there is no nanoparticle there, you can see even 10,000 10 to the power of 5 molecules and over 400 seconds accumulated, the spectra looks very weak. But here you can see one molecule, 0.05 seconds, you get a very beautiful spectra. So this is what is very important when we actually look at this system. Now, 1 minute, I just had a timer going on so that I don't overshoot the time. So basically this is what we use mostly for drug discovery. Now, one of the places where drug discovery needs is screening of the drug, and also finding out new variants of the drug, whether it will work or not. For this, most of the times it is the X-ray crystallography, and you use molecular dynamic simulations to actually find out the binding sites where the molecule binds. The first known thing was done which got a Nobel Prize, was done in 1962 for myoglobin. And this one actually is not going to be replaced by Raman. So what we have actually realized is that now we have AlphaFold 2, which is from the Google, which can also predict the structure of the protein. So now Raman is, where does it come? So we actually believe that we can actually use Raman in complementary to these two things for finding out where the drug goes and binds. And if we know where it binds, then we can actually find out what kind of changes would actually help in getting a better drug out of this system. Or the biochemistry needs to be done less. This becomes like a screening tool. Now, Raman has a very interesting property that it looks at the peptide bond, that is C double bond ONH bond. And this C double bond ONH actually has this unique vibrations. And these unique vibrations are called amide 1, amide 2, and amide 3. And these vibrations are very unique because they are the places where the hydrogen bond happens, and because of which this folding happens. So the secondary structure like alpha helix, beta sheet, beta turn, or random coil, or antiparallel beta sheets can be actually found out from the amide peak. So the amide 1 peak actually can be deconvoluted, and for each of these proteins, there will be a specific kind of thing. And the intensity tells you how much is of each of these components. And we can actually find out. So here you can see for stratum cornum, it is basically having alpha helix is the maximum. Beta sheets are there, random coils are there, and antiparallel beta sheets are there. So you can see that this is something. So I take one example to prove that we can actually use Raman. And one of the thing is that you have kinases, and Aurora kinase, Aurora A, B, and C are involved in cell division. And you will realize Aurora A actually helps in forming the spindles, and Aurora B actually helps the chromosomes to align itself in the equatorial plane. And Aurora C actually works in combination with Aurora A and B to actually help in doing the cell divisions properly. It is well known that all the tumors, Aurora A is overexpressed, and because of that, you have non-uniform spindle formation, and that leads to actually daughter cells being not viable, and also cancerous systems which will happen, and aneuploidy happens. So it is always a target. Aurora A is a target for cancer therapy for this things. Now, when we were actually working with this with my colleague at JNCSR Tapas, and my student currently, he is a faculty at IIT Delhi, we were looking at the Aurora A and Aurora B. And very importantly, when you look at the structural alignment and the sequence alignment, you see that it is very much similar. And you can realize that any drug which should bind to this should bind to this. So that means catalytic site is similar. So that is an ATP binding site. This is the catalytic site, and this is the catalytic site here. it is ATP binding, and this will be the same. And Raman, you can see more or less for a novice, it looks identical spectra. And you can realize that if I zoom it up, it shows that there is a strong peak around 1616 for amide 1, which says it is from beta sheets. And this is the structure of the Aurora A or Aurora B. And you can realize that it is sitting on top of the nanoparticle with the beta sheets. Now you do the assay, and then you can see that with the philidipine, which is a hypertension drug, which actually is out of AstraZeneca's place, this is supposed to have a inhibition of Aurora A specifically, not for Aurora B. So this means this is a very interesting molecule to understand where does it bind. If you look at a generic drug, which is nitrodipine for the hypertension, it has a nitro group instead of a Cl present here. So this is a hydrophobic region and it gets locked in this position. This has a rotational degree of freedom. Hence, this doesn't work at all in activating. So this is how we can actually understand. Now, you can realize in this is your catalytic site. So this is the type 2 inhibitors which go here and type 1 inhibitors which go at the surface, at the hinge region. Now, you realize as soon as you put philidipine, you start seeing that suddenly from a beta sheet, it is going to an alpha helical structure. So what we realized for Aurora A is that because of the chlorine, which is hydrophobic, it shifts itself from the beta sheet region to an alpha helical region. That means this philidipine is actually binding at the type 1 inhibitor site. While when we look at the Aurora B, it doesn't bind at all. So that means the binding site is this. Now we do molecular dynamic simulation and we were able to see that the docking and molecular dynamic simulation shows that it actually binds to this pocket, which is actually having attachment to phenylalanine, isoleucine and tyrosine. And if you do the mutagenesis at this point, and you immediately see the wild type, there is an inhibition, while the inhibition is lifted the moment you do the mutagenesis of this one. And that means this has been proved to be the binding site of this. Now, why is it actually binding only specifically to Aurora A, not to Aurora B? Because in the Aurora A, there is an extra histidine, which gives you a 3 to 4 Armstrong capacity to increase. And that is the reason for the selectivity of the Aurora A inhibition by this philidipine. Now, with this knowledge, we can actually go ahead and do a lot of studies on which molecules to actually work with. All of you would have realized philidipine actually changes your blood pressure. So naturally, a normal person cannot be given this because his blood pressure would change if he is actually trying to take it for tumor. So with that, I would come to an end. I hope I have actually given you an impression of how Raman works and how Raman can be used for drug discovery. Thank you for your attention and happy to be here, and thank you for the opportunity.
Thank you, sir, for that illuminating and scholarly keynote address. You have taken us from what happens when light falls on material, Raman scattering, Raman spectrophotometer, and also how the vibrations of molecules will become fingerprints of compounds and how we can characterize them, and even how we can detect pesticides in vegetables and fruits, and also for the identification of fake drugs, and also how nanoparticles can help in drug discovery. Sir, you have beautifully linked the legacy of Sir C.V. Raman with contemporary drug discovery research. Thank you, sir. Now, we are moving to distinguished talk from our guest of honor. It's a great honor for me to welcome our esteemed guest of honor, Dr. Sri Gobi, Chairman and Managing Director of Molecules Group, Thrissur, who has been guiding a force and a visionary in the pharmaceutical industry. Now, I invite Dr. David Paul, Assistant Professor, NIPA, Kolkata, to kindly introduce our distinguished guest of honor. Over to David Paul, sir.
Thank you, Julie ma'am. Good afternoon, one and all. Distinguished dignitaries, respected faculty members, and my dear students. It's my great privilege to introduce our guest of honor, Dr. Sriraj Gobi, an accomplished scientist, innovator, and entrepreneur with over 17 years of excellence in translational research and product development. Dr. Sriraj holds PhDs from Dansk University of Technology and Deakin University of Australia in liposomal technology. Dr. Gobi has 200 plus research publication, holds multiple patents, and a distinguished fellow of Royal Society of Chemistry as well as fellow of the Royal Society of Arts. He is also recognized among the world top 2% scientists by Elsevier Stanford for the consecutive 5 years. He is the founder and managing director of Atomic Innovations and Molecules Biolab, chief executive officer of ACNO Formulation, and he is also served as a government nominated director of Kerala Life Science Park. With a unique blend of academic and industrial expertise, Dr. Sriraj served as an advisor to numerous companies and also visiting professor at prestigious universities. Dr. Sriraj is also the recipient of TIE Kerala Innovator of the Year 2025, CII National Award for the Best Intellectual Property in India. With a blend of numerous accolades, we are truly honored to have such a visionary with us today. Welcome, sir, to this event. Sir, please.
Thank you very much. Have a good afternoon to all. So let me share my screen. Can you see my presentation?
Sir, you are sharing your screen directly, not PPT.
Yeah, now it's visible.
No.
I have shared that. I don't know. Why is it not coming?
Sir, you just go for share screen, sir, first. I will guide you, sir. Possibly it will work out. Share screen, sir. Then the screen. Then now go to PPT, sir, PowerPoint, which is open and selected.
Yeah, it's okay now, right?
Give a slideshow mode, sir. Yes, it's fine. Now it's fine.
Okay. Thank you. Sorry for the inconvenience.
Julie, madam, is it fine, no, ma'am?
It's a
Yes,
fine.
sir, it is fine. It's fine
Okay,
now.
okay. Thank you.
Okay. Again, good afternoon to all. So, as you all know, today is National Science Day, and the theme is woman scientist. So I have conceived that this year theme And my topic for today's discussion is Women Scientists as Innovation Leaders, Transforming Research to Impact. So, as you all know, the present scenario, the science scenario in India, India produces close to 50%, for almost 43% women STM graduates, and which is a strong participation in life science and biotechnology. And as you are seeing right now, the women PhDs in biological sciences, almost all disciplines are increasing, but especially for the research graduates, which is coming out of biological sciences, are rising. And but when you compare that number, and I represent here as an industry, so when you compare the number which is graduating and the representation in our industrial sector, is there's a huge gap. And this gap is not about talents, because as you can see, more number of women are coming into the research degrees, and they are passing out with excellent publications and stuff like that. But the gap is not about talent, it is about the ecosystem that we have created here, as you all agree already. And now, the translational gap, the academic research into the industrialization. There are high quality academic publications, but this limited conversion to a IP and a scalable products. And our ecosystem always look for or take grants, often a publication driven, not a product driven, or how many IPs has gone into a scalable product that is not visibly implemented in our system. It's almost how many publications you have taken, you have produced, and what's the impact. I'm not saying it is not a good idea, but still there has to be some clear advantages given to someone who has taken the research into an IP and into a scalable innovation. And this need a shift from research output to research impact. It's not just merely research output. There has to be a leverage on research impact. And to give my own translational story is about the creation of molecules biolabs, where we have created from the scratch. And it was merely our bench type academic research on liposomal research and liposomal drug delivery systems. And right now, where we have close to an export history of close to 48 countries. And we are pioneers in liposomal research, which has taken a few years to translate the academic research into a translational research and into a scalable operation, which is now a self-sustained, industrial research. And not that we have not done publications, we also did numerous amount of publications. But the thing is that there are two set of research where there are some things which only it will concise to only academic research and ending up with a publication, which is very good. But there are another set of research which can be taken into a scalable operation, which will indirectly fund the basic research. Basic research is always important, but application oriented and academic scalable operation is also essential for our industry to grow. And this is our global footprint, where we have 3 manufacturing unit, 3 R&D units, and this is a pretty old presentation. I think we right now we have more than 50 scientists and 48 global sales channels and numerous publications. And when I speak about scientists, our team, I think I will come to that later, having more than 60 percentage women scientists, very talented women scientists. And our ecosystem model is an applied and commercialization focused research, which is IP-driven innovation, integrated R&D, validation, and then going into manufacturing. And this is an inclusive and performance-driven culture, which is not always possible with an academic research, but it is always possible to conceive from academic into an ecosystem where then only the academic research also will flourish, where you scale up whatever product or technology or drug delivery tools which is developed in academy, and then taken into a transnational path where you end up with IP. And so that's one thing which India always should look into. As I think Professor Jayashekhar was mentioning about the India's contribution to the R&D, we should invest more into R&D and IP. And IP is something which will give more value even rather than merely a contract manufacturing hub, which used to be the case for some years, but there is a shift. For example, our technology itself is a perfect example. India used to import liposomal drug delivery systems from Europe, but right now we satisfy almost 60% of liposomal supplements needs for the Western community in the supplement market. So which means that the IP here lies here, where we have more negotiation power, and also it will further strengthen our ecosystem, which is science and technology driven. And again, coming to the scientific workforce, what we have implemented, and I don't think I can boast of that we have a particular policy that we will invite only women scientists. It doesn't happen that way. The 60% of the scientists across our, we have several ecosystem, allied companies and stuff like that. 60% of the scientists are women scientists, and they lead the formulation R&D. They drive the analytical validation, and they are managing the technology transfer to other companies. And the leadership emerged through opportunity and the merit. And uh, what are the observation from my own experience when we take up women scientists as the leadership role, high process discipline, and strong documentation culture, which sometimes lack in the it has, I think, from my own experience, some sort of strong documentation culture for them, and long-term project commitment and resilience into translational execution and innovation becomes a structured execution when they are in the powerful post. And uh, why translational biotechnology enables women leaders? So the perfect example, I think the previous speakers already mentioned notable women scientists or leaders in the sector. It need not be always scientists, it could be also leaders. Like, for example, the perfect example is Kiran Majumdar Shah, who has made the biotechnology hub with a company like Biocon. So a detail-oriented and interdisciplinary culture should be in place, documentation and regulatory intensive, and it's a interactive product development cycle and high impact on healthcare and society. And now let's go to the basics, and from research to market impact. How do we achieve that research to the market impact? Uh, so we do the research, and then most of the time the academic research stops with the publication impact. So we take up to that level, but we never go to a prototype level. And it's not merely a prototype. Then prototype need to validate, and then go for an IP, intellectual property, and then go to the real manufacturing and to the market. So this is a very long cycle where once you are in the market for about few years, which means that the research has continuously validated. It's not just about peer review. It's about how you are creating some research into a really marketable innovation. And women scientists actively lead multiple steps in this chain, and impact becomes measurable when research becomes scalable. And this is very important. Otherwise, we cannot, the impact is not there when you have no scalable product or process in place. And that's the reason why it is very important. And I am going back to the today's theme of for the Vikasit Bharat. What is the roadmap? So encourage women in translational and industrial leadership, which is not that evident in today's lifestyle or the ecosystem, because we are not seeing much women in the industrial leadership place. And support women-led biotech enterprises, align funding with commercialization goals, and recognize impact beyond publication. That's a very key important steps. We only recognize not that that is not not needed. Publication is essential for a science to grow, but just not recognize that alone, but recognize also an impact beyond publications. And it's a closing thought. Science transforms a nation only when research becomes an impact. US is the best example for that, and women scientists are central to that transformation. Inclusive innovation is the foundation of Vikasit Bharat. And again, once again, thank you for inviting me for this special lecture series, and wish you very best and a happy National Science Day. Thank you.
Thank you, ma'am.
Thank you, sir, for sharing your valuable insights. So the sir has told to us how the translation gap and his own translational story from academic research to scalable industrial research. It was truly inspiring. As he said, the importance of scalable operations, inclusive and performance-driven culture in pharma industry. And he also highlighted the importance of women scientists, how they can contribute exemplified manner in documentation and leadership. And it was a very, very good example of Kiran Majed Shah of Biocom. So research impact become measurable only when it becomes scalable. So we hope from your speech, sir, or after hearing you too, sir, women become more powerful and play key roles in this industry area in future. Thank you, sir, for your valuable notes. Sir, my slide is visible, sir?
No, not yet.
Prashant, sir, can you just see whether it was...
Julie, once again open, madam.
Okay.
Let's make it view slide.
Yes.
Click on view slide. View slide. Okay. Make into slideshow.
So now we move to invited young scientist talk, which highlights emerging talent in scientific research. It's my pleasure to introduce Dr. Ashwati R. Devan, DST Inspire Fellow from Amrita School of Pharmacy, Ernakulam. Dr. Ashwati completed her PhD in Pharmaceutical Sciences at Amrita School of Pharmacy, Kochi, under the guidance of Dr. Lakshmi R. Nath. Her doctoral research focused on flavonoid-based combination therapy for hepatocellular carcinoma. She holds both B.Pharm and M.Pharm degrees from College of Pharmaceutical Sciences, Government Medical College, Thiruvananthapuram, and secured first rank in M.Pharm pharmacology. In recognition of her research potential, she was awarded the prestigious DST Inspire Fellowship in 2020 for her PhD research. During her doctoral tenure, Dr. Ashwati has authored 21 scientific publications in Scopus Index high impact journals, reflecting her strong research contributions. She is also a recipient of several accolades, including the Biomedical Systems Endowment Award for Best Oral Presentation at the 38th Kerala Science Congress, the Best Oral Presentation Award at the 7th International Conference on Nutraceuticals and Chronic Diseases, and second prize at ANRF-sponsored National Conference held at Al Shifa College of Pharmacy. Now, I welcome Dr. Ashwati to take over the stage. Over to Ashwati, ma'am.
Thank you, ma'am.
So, ma'am, you have the slide to share?
Yes, yes.
Okay, so I will leave.
Ma'am, is it okay, ma'am?
Yes, it is visible.
Okay. So good afternoon, everyone. Ma'am, thank you for the introduction. I am Dr. Ashwati Ardevan, and I am here presenting the major findings of my doctoral research, which I have done under the guidance of Dr. Lakshmi Arnath at Amrita School of Pharmacy, Kochi. And in our study, we found that the dietary flavonoid camphorol potentiates sorafenib response and reduces the treatment-associated toxicity in hepatocellular carcinoma. So hepatocellular carcinoma is the most common form of liver cancer. In advanced stage, drug therapy is the main treatment, in which sorafenib is the first approved first-line therapy, and it still remains as the globally accepted standard care for HCC. However, at an advanced stage of the disease, majority of the patients cannot tolerate these toxic side effects that either lead to therapy discontinuation or dose reduction. And another problem is the development of sorafenib resistance. So several studies report that the abrupt activation of the transforming growth factor beta is associated with a poor prognosis in HCC. Therefore, the combination of a TGF-beta inhibitor such as galunisertib with sorafenib improve the overall survival in phase 2 clinical trial. But unfortunately, the development of cardiac toxicity denied clinical approval. So this highlights the need of some safe, effective alternative TGF-beta inhibitor, particularly in combination with sorafenib. So our strategy is a phytochemical mediated chemosensitization from a panel of 20 different flavonoid molecules. Using the molecular docking, we found that camphorol is having the best interaction with the TGF-beta receptor 1 kinase as compared with the standard drug galunisertib. So camphorol is a well-known flavonoid molecule abundantly distributed in fruits and leafy vegetables. So literature evidence also suggests that the camphorol can target the TGF-beta signaling in multiple disease models. And importantly, the majority of the biological sources of the camphorol are generally recognized as safe by US FDA, and also it's a cardiac protectant. So based on these evidence, our plan is to evaluate camphorol mediated sensitization of sorafenib via targeting the TGF-beta pathway in HCC. So first objective is to optimize the synergistic concentration of camphorol-sorafenib combination and compare the effect with that of the galunisertib-sorafenib combination. First, we identify the individual cytotoxicity of both camphorol and sorafenib in 3 HCC cell lines, N1S1, HUH7 and HG2. And based on this IC50 value, we selected the subtoxic concentration of the sorafenib, that is from 0.5 to 2.5 micromolar, and combined with the suboptimal concentrations of the camphorol, that is from 2.5 to 100 micromolar. So we tried around 30 different combination ratio. And in order to quantify the nature of the interaction, whether it is additive, antagonism, or synergism, we used different mathematical models. So in the combination index model, if the value is less than 1, that indicates synergism. In another 2 model, if the score is greater than 10, that indicates synergism. mechanism. So we tried 5 sets of ratios in 3 HCC cell lines, and we can see that among the 5 panel, this last panel is showing some synergistic cytotoxicity, which is clearly evident from the low combination index value and also a synergy score of greater than 10. So this panel, that is sorafenib 2.5 micromolar combined with the 2.5 to 100 micromolar of the camphorol exert a very promising synergistic cytotoxicity in HCC cell lines, which is evident from the combination index plot where the points are below 1 and also in the isobologram where the points are below the line of additivity. So next we compare this effect with that of the galurisertib sorafenib combination. So the purple color camphorol sorafenib, particularly at a higher combination ratio, exert a superior efficacy than the galurisertib sorafenib combination. Now, in order to prove the TGF-beta mediated action, we evaluated the effect in presence of the TGF-beta. The TGF-beta treatment increased the proliferation and also induced epithelial to mesenchymal transition in the cell. But in presence of the TGF-beta, the camphorol sorafenib combination synergistically inhibited the growth, where the effect was significantly superior than the galurisertib sorafenib combination. The TGF-beta treatment increased the colony formation, which is significantly inhibited by the combination. So the combination induced apoptosis and also it inhibited the TGF-beta mediated migration of the HG2 cell. Now, as a proof of the mechanism, we evaluated the downstream target of this pathway, that is SMART2 phosphorylation. So in the TGF-beta treated group, there is a high level of the phosphosmart. So that indicates the active signaling in the cell, where the combination significantly inhibited the SMART phosphorylation with a comparable efficacy with the galurisertib sorafenib combination. So this is our in vitro evidence, and we found that the TGF-beta treatment can induce the proliferation, induce epithelial mesenchymal transition and the migration in the HCC cell, where it was synergistically inhibited by the camphorol sorafenib combination with a superior efficacy over the galurisertib sorafenib combination. So the next objective is regarding the pharmacological safety. First is the acute oral toxicity evaluation as per the OECD 423. So here in STRAT, we used sorafenib 20 mg per kg and combined with a very high dose of the camphorol, that is 2000 mg per kg. After single oral administration, the animal is observed for 14 days, and during the treatment period, there was no abnormal behavior or mortality that indicate the LD50 of the camphorol is greater than 2000 mg per kg when it is given in combination with the 20 mg per kg of the sorafenib. In addition to that, the hematological profile, the liver function, renal function, and the cardiac function, along with the histopathological validation, confirms an acceptable safety at a very high dose range in acute exposure. So now coming to the repeat oral toxicity, that is subacute toxicity evaluation by OECD 407. So here we combined 100 mg per kg of the camphorol with 20 mg per kg of the sorafenib, and the drug is administered continuously for 28 days in both male and female animals. So the treatment was well tolerated. There was no mortality, abnormal behavior, or body weight loss. And furthermore, the normal hematological profile, liver function, renal function, and cardiac function, along with the histopathological validation, confirms the acceptable pharmacological safety of this combination on repeated use. So next is an important objective is regarding the in vivo efficacy of the combination. First, we used the immunosuppressed model, that is HG2 xenograft model in athymic nude mice, where the tumor induction was performed by the subcutaneous injection of HG2 cell, which is mixed with the metrigel into the flank region of the mice. And the tumor induction was confirmed by day 7, and we started the treatment orally when the tumor volume is around 50 to 100 millimeter cubes. So we have 5 groups. One is the vehicle control, second is the combination, camphorol 100 mg per kg and sorafenib 20 mg per kg. So next is the individual group, that is sorafenib 20, camphorol 100. So we also included a therapeutic equivalent dose of the sorafenib, that is sorafenib 40 mg per kg. Weekly three treatment for 4 weeks. We can see that after this 1 month of the treatment, camphorol sorafenib combination significantly inhibited the growth of the HG2 xenograft tumor, where the effect was found to be superior than the sorafenib 40 mg per kg. So in a detailed view, in the control tumor, the volume was around 1800. So in the combination, it's around 300. That means a 5-fold reduction. But in the individual group, sorafenib 20 and camphorol 100, it exerts a 1.5 and 1.2-fold reduction. So a 5-fold reduction with the combination. So here also confirming the synergistic anti-tumor efficacy of this combination in the animal model, which is further validated histopathologically. Now, in order to analyze the cell death or the apoptosis induction in the tumor tissue, we performed the tunnel staining. So the brown colored cell that indicate dead cell or apoptotic cells, we can see that in the combination treated group, there is significantly more number of apoptotic cell as compared with the individual therapy. So that confirming the promising anti-tumor effect of this combination as compared with the individual drug. So next is an immunocompetent model. So that is N1S1 syngeneic orthotopic model in SD rat, where the tumor induction was performed by the orthotopic injection of N1S1 cell into the left lobe of the liver using a laparotomy surgery. And the tumor induction was confirmed by day 7, and we started the treatment when the volume is around 100 to 150 millimeter cube. The same group and the same schedule, but here, as the tumor is formed inside our body, we need an imaging tool for the continuous monitoring of the tumor. In most of the studies, they used either MRI or CT, which is very expensive. But for this study, we optimized ultrasound imaging as a cost-effective tool, and we can see that in the ultrasound image, in the tumor control, so there is an exponential growth of the tumor. So that indicate the aggressive or rapid growth of the tumor. But we can see that in the combination, it remarkably inhibited the growth of this N1S1 tumor, leaving just a scar-like portion at the tip of the liver, where the effect was comparable with the therapeutic equivalent dose of the sorafenib. So now in a detailed review, in the control, the volume was around 4,000 millimeter cube. That indicate the very aggressive growth of the tumor, where the combination reduced the tumor volume to around 30 millimeter cube. So that confirmed the excellent anti-tumor potential of this combination therapy, which is further validated histopathologically. Now, as a proof of the mechanism, mainly the TGF-beta can trigger epithelial mesenchymal transition and promote metastasis, and also it increases the proliferation of the tumor cell and also suppress the anti-tumor immunity. So therefore, in order to prove our mechanism, we analyzed these 4 markers in the tumor tissue using the immunohistochemistry. We can see that E-cadherin is an epithelial marker and vimentin is a mesenchymal marker. So in the tumor control, so there is a suppression of the E-cadherin and also the overexpression of the vimentin. So that indicate the epithelial to mesenchymal transition in the control tumor, where the combination, it shows the restoration of E-cadherin and also suppression of the vimentin. That indicate our combination is effectively inhibiting the epithelial to mesenchymal transition. Similarly, Ki67 is a proliferation marker which is significantly inhibited in the combination treated group. And also the overexpressed PD-L1 in the tumor control indicate the suppressed anti-tumor immunity, where it was significantly inhibited in the combination treated group. So this confirmed the TGF-beta mediated action of this combination. Now another is important regarding the superior safety of this combination. And during the treatment period, in sorafenib 40 treated group, so there is an abnormal elevation of SGOT that can indication of its liver toxicity. But the camphorol sorafenib combination maintained a normal level of liver function in both SGOT and SGPT during the treatment period. And importantly, the sorafenib 40 treated group shows an abnormal elevation of the CKMB, that's a cardiac toxicity marker. But in the combination treated group, that maintained a normal level of the CKMB. So this confirms the superior efficacy of the camphor efficacy as well as the safety of the camphorol sorafenib combination as compared with the therapeutic dose of the sorafenib. So with all these preclinical evidence, we are reporting a novel, well-tolerated combination therapy that can improve the efficacy as well as the safety of sorafenib in hepatocellular carcinoma. And also, we validated camphorol as a safe and effective chemosensitizer, which definitely need further clinical evaluation. So in the light of this promising evidences, our department, our lab, as well as our institution is trying to extend the study to a translational level. So therefore, additional experiments and discussions are ongoing to submit an investigational new drug application for initiating the human trials of this combination, particularly with the camphorol. So this The study is financially supported by the DST Inspire Fellowship and also Amrita Vishwavidyapeetham Seed Grant. And in recognition of this work, I received the Biomes Medical System Endowment Award for the Best Oral Presentation in Biotechnology category at the 38th Kerala Science Congress, and also first prize in oral presentation in the 7th International Conference on Nutraceuticals and Chronic Disease, and also secured second prize in an ANRF sponsored national conference. And during my tenure at Amrita School of Pharmacy, so I authored 21 scientific publications in Scopus Indexed High Impact Journal, where the 6 publications are directly derived from my PhD work. And coming to the acknowledgement, first and foremost, I am immensely grateful to our Chancellor, Amma, for all the divine grace and blessing that helped me for the successful completion of my doctorate study. And I am grateful to our Associate Provost, Dr. Shantikumar V. Nair, our Principal, Dr. Sabidayam, and my guide, Dr. Lakshmi Arnad, because their insightful suggestions and critical evaluation refined this work to this level. And I'm grateful. I'm thanking my Department of Pharmacognosy, and the entire Amrita School of Pharmacy for all the support. And definitely, I'm thanking Indian Pharmaceutical Association Kerala branch for this great opportunity. And thank you so much.
Thank you, Dr. Ashwati, for an excellent and well-presented research talk.
Thank you, ma'am.
As a woman, I feel proud of you.
Thank you, ma'am.
Now, we move to the second invited young scientist talk, which continues our focus on emerging research and innovation in pharmaceutical sciences. It is my privilege to introduce Dr. Dilin PM, an accomplished academician and researcher in the field of pharmaceutical sciences. Dr. Dilin is currently serving as an associate professor at Dr. Nobel's College of Pharmacy, Wayanad. He completed his PhD from Anna University, Chennai, and earned both his B.Pharm and M.Pharm degrees from Government College of Pharmaceutical Sciences, Government Medical College, Kozhikode. His research interests span peptide drug delivery system, nano drug delivery systems, cosmeceuticals, cosmetic science, reflecting a strong interdisciplinary approach to pharmaceutical research. Dr. Dilin has contributed significantly to the scientific community through publications in international peer-reviewed journals, and also has authored books and book chapters in his areas of expertise. He has filed 2 patents demonstrating his commitment to innovation and translational research. And he is widely respected for his dedication to teaching, research excellence, and mentoring students in advanced pharmaceutical sciences. I now invite Dr. Dilin PM to present his research titled Formulation and Characterization of lactoferricin beta-loaded nano-ml gel and fisetin-loaded neosomal gel for effective melanoma treatment. Over to Dilin, sir.
Thank you, ma'am. Am I audible, ma'am?
Yes, sir. Just you want to share, I will just stop sharing.
Making slideshow mode. Right.
Thank you, ma'am. Good afternoon, one and all. I'm very happy to present my research work as part of National Science Day webinar conducted by IPA Kerala State Branch on topic formulation and characterization of lactoferricin B loaded nano ml gel and fisetin loaded neosomal gel for effective melanoma treatment. I'm Dr. Dilip PM, Associate Professor, Dr. Nobel's College of Pharmacy, Wayanad. This is the part of my doctoral research under the guidance of Dr. K. Rukmani, Director in IPA Hajipur. As we all know, cancer is a disease in which abnormal cells divided uncontrollably and destroyed by tissues. Mainly, skin cancers are divided into 2, melanoma and non-melanoma cancers. And melanoma cancer, non-melanoma cancer, which include basal cell carcinoma and squamous cell carcinoma. Melanoma cancer is the most lethal type of skin cancer and the most common cancer all over the world. Poor prognosis and survival if not treated early. The conventional treatment strategies, which include surgery, chemotherapy, radiation, immunotherapy, laser ablation, and regional hyperthermia. The recent treatment strategies, which shows a better cure rate with the topical delivery of the drug. Here we selected topical drug delivery because of non-invasiveness, avoidance of hepatic and GI metabolism, prolonged delivery, and good acceptance by the patient. The recent treatment strategies, which shows better cure rate with the topical administration. This is one of the examples for the same by using imiquimod. There will be a chances of localization of synthetic drug only to the outermost layer of the skin, which causes severe skin irritation, burning sensation, etc. Cytotoxic concentration is failed to achieve at a deepermost layer. That's why we selected two natural molecules for the preparation of various formulation, which include animal-derived peptide, lactoferrin B, and plant-derived flavon physicist. Even though I think the slides are not changed. My first work is development of novel lactoferrin-B-loaded nanoemulsion for the effective treatment of melanoma. The reason behind the selection of lactoferrin is because of its high content of cationic and hydrophobic amino acid, which like arginine and tryptophan, which will interact with the anionic cell membranes, which is present in the cancer. The mechanism behind the anti-cancer, which includes induction of cell necrosis and cell apoptosis. Even though the peptide are excellent candidate for cancer treatment, there are some barriers in the which include hydrophilic nature, high molecular weight, hydrophobicity of spermiconium and proteolytic enzyme, which is present in the skin, which will destroy the peptide. Why we have selected nanoemulsion? Here we have prepared water nanoemulsion because of it's a hydrophobic continuous phase that covers the hydrophilic surface of the protein peptide, and which will overcome the 500 Dalton rule for the skin permeation of hydrophilic molecules. The oil titration techniques, which is used for the preparation of emulsion, allows the final emulsion with droplet size in the nanoscale matrix. Skin penetration assisted by the surfactant and oil, which will helps to more permeation into the skin. All the problems are overcome by this preparation of nanoemulsion. Then here, first objective is the development of HPLC method for the lactoferrin B. Till though there will be no method developed for lactoferrin. So I first developed the HPLC method for lactoferrin B. The method was validated for linearity, method precision, limit of detection, limit of quantification, inter-day variability, robustness, and solution stability. This is the typical chromatogram for lactoferrin B. For the preparation of lactoferrin and nanoemulsion, water titration method is used by mixing aqueous phase of lactoferrin with S-mix, twin 20 and span 80. Here, isopropyl myristate is used as the oil for the preparation of lactoferrin emulsion. By changing the ratio of surfactant mixture and oil, 4 series of formulation were prepared and phase diagram was prepared. This is the four phase diagram, which shows the range of nanoemulsion which is prepared. The selected 4 formulation were optimized for globule size and PDI and drug content. From these 60 formulation, 26 formulation we selected based on the clarity. Then from this 26 formulation, 4 formulation based on the stability of after 2 weeks were selected. The formulation FA2 was selected out of 4 formulation due to very low globule size and PDI value and the highest drug content. The formulation FA2 was further characterized for various parameters. This is the parameters which is observed after the preparation of FA2. The functional group, rheological parameters, surface morphology, and the globule size were optimized. After that, the ex vivo skin permeation studies were performed. Pure lactoferrin B exhibited a negligible rate of drug release after 24 hours. This is because of hydrophilicity and high molecular weight of the peptide, which is a barrier in the skin permeation. But nanoemulsion exhibit a very slow and sustained rate of release, and the accumulated rate of 83.17 was observed even after 24 hours. This suggests that remaining concentration will be retained within the dermal layer, suitable for topical treatment of melanoma. The result in brief, which shows globule size obtained for the nanoemulsion is about 53.95, which will enhance the stability with less chances of coalescence, and the globule morphology, which will shows evenly distributed, discreted, uniform spherical shape without any aggregation, and pH is compatible with the skin, and the transmittance, which shows transparent formulation, complies with the definition of nanoemulsion. My second objective is development of physitin-loaded neosome for the effective treatment of melanoma. Physitin is a phytoconstituent which is emerging nutraceutical and therapeutic application, which is used for anti-cancer, anti-diabetic, neuroprotective, angiogenic, and anti-arthritic effect. Because of its poor aqueous solubility, high lipophilicity, and low oral bioavailability, we need a nano formulation for the further evaluation and treatment strategies. This is the graphical representation of preparation of physitin-loaded neosome by probe sonation method by using physitin cholesterol span 60 and phosphate buffer. Here we prepared various ratios of formulation F1, F2, F3 by using cholesterol span 60 in the ratio of 1:0.5, 1:1, and 1:1.5. And further, these formulations are characterized for vesicular size, surface charge, engraftment efficiency, in vitro drug release, thermal behavior, drug excipient compatibility, and surface morphology. Here, neosome was prepared at various ratios and determined the particle size, PDI, CT potential. From this, F2 was further selected for further evaluation, and the optimized F2 furtherly we are observed by using various amplitude and sonation time and temperature. From this 18 formulation, the C6 was prepared with cholesterol span 60 ratio 1:1, sonicated with an amplitude 35% for 5 minutes at 63 degrees Celsius, was selected as a highly stable uniform nano size optimized for final formulation. Hence, the finalized formulation C6 is further denoted as F2 in the entire study. And this prepared F2 formulation was characterized for time analysis. The morphological observation revealed that neosome were homogeneous and monodispersed. No apparent aggregate formation was observed among the neosome. And the DSC analysis also shows the peak of pure drug in the thermogram of formulation confirmed the successful formation of drug encapsulated neosome. And FTA spectrum demonstrate an absence of characteristic peaks of physitin in the development formulation, suggests the efficient encapsulation of the pure drug in the neosome vesicle and assured the drug excipient compatibility. XRD spectra, which shows physitin-loaded nanovesicle, doesn't show the peak of pure physitin. That indicates the encapsulation of physitin in the nanovesicle. The crystalline nature of, as per XRD pattern, show amorphization of physitin after the physitin-loaded nanovesicle. This was resulting in consistent 4 nanovesicle formulation. Further, ex vivo skin permeation, which shows the result as a controlled permeation of the drug from the formulation F2 throughout the period of 24 hours when compared to pure drug. And nearly 20% of the drug is permeated from the formulation even after 8 hours of diffusion. This suggests that the remaining percentage of the drug is retained within the skin layer and released slowly due to the reservoir action of neosome within the skin. My next objective was for enhancing the skin retention and ease of application. The prepared neosome and nanomulsion were dispersed in a suitable gel matrix to form neosomal gel and nanomul gel and evaluated for in vivo anti-cancer activity. The advantages of this prepared nanoemul gel and neosome were ensures the sufficient mucoadhesive property, hydrate the skin to open up the pores that facilitate the easy penetration, enhanced bioavailability, prolonged skin retention, and control the release over a prolonged time. The fabrication of polymeric composite were of the preparation of nanoemulsion done with the lignin pectin gel at the various ratios. For the preparation of gel-based polymer like a lignin pectin gel were taken at different ratios and characterized for particle size, PDI, zeta potential, surface morphology, and a rheological evaluation. Out of which 1:1:2 and 1:3 ratio of LPG and LPT correspondingly was selected for further analysis and determine the FTR and DSC methods. In both LP and LPG composite, the thermal stability is better to individual polymer which promote their application toward the development of suitable topical hydrogel. But when considering the adequate gel strength, LPG is selected for loading of nanoemulsion and neosome when compared to LP. So the lignin pectin gel and composite was selected as optimized gel base. And the in vitro characterization of nanoemulsion and neosomal gel were observed for physical appearance, pH, and viscosity. And the in vivo skin irritation studies were conducted. All the formulations were found to be non-irritant to the skin, which will indicate the safety of developed formulations. And finally, in vivo anti-cancer activity of developed formulation were performed. The melanoma cancer was induced by using DMBA protonoid. 8 groups are there for the further studies of animal studies. Tumor incidence and tumor burden, which shows among the experimental formulation, the treatment with fisetin neosomal gel and lactofuricin nanoemulsion showed significant reduction in the both tumor incidence and burden, which is comparable with the standard treatment group. Hematological analysis, total protein evaluation also performed. Enzymatic and non-enzymatic antioxidant studies are also performed. And finally, the morphological assessment, which shows DMBA only group exhibit distinct signs of skin carcinoma, including thickened epidermal layer, hyperkeratosis, and presence of abnormal and atypical cell growth. Groups treated with the standard drug, neosomal gel, and nanoemulsion exhibit reduced tumor formation and improved skin morphology with fewer lesion and less cellular disorganization. Histopathology study also shows that DMBA alone group, the epidermis exhibit neoplastic hyperplasmia with edematous collagenized dermis and demonstrate signs of chronic inflammation. The treatment with the standard drug, neosomal gel, and nanoemulsion groups resembles the control in its papillomatous hyperplasmia, epithelial, and edematous subdermal layer, which suggests protection against DMBA-induced alteration. Into the conclusion, the developed novel lactofuricin B-loaded watery novel nanoemulsion and fisetin-loaded neosomal gel were found to be a better formulation compared to free drug upon topical application due to its nano size and liquidic nature. Moreover, the loading of nanoemulsion and neosome into a bioadhesive polymeric nanogel enhanced the effectiveness of formulation by hydrating the skin cell, providing more resistance time to penetrate, protect from the external environment. So the developed formulation suggests that a prominent therapeutic agent for topical treatment of melanoma cancer. These are my reference. And I would like to express my deepest gratitude to my supervisor, Dr. K. Rukmani ma'am, for her valuable guidance and encouragement. I'm grateful to my beloved principal, Dr. Radhakrishnan, and the management of Dr. Nobel's College of Pharmacy, Wayanad. And I would like to extend my deepest gratitude to the IPA Kerala State Branch, and I extend my sincere thanks to the funding agencies of NFTD and FBPDST for their support, which made this research possible. Thank you.
Thank you, sir, for the informative and impactful presentation. Now, we have a Google feedback form that is posted in the chat box. So please fill it and post back. So you will get the participation certificate. Is the slide is visible?
Yeah.
Oh. Sir, is this slide is visible?
Now it has gone. No
No.
problem. Yeah, it's
Madam, please keep it like that. Okay.
Okay, fine. So as we come to conclusion of today's enriching program, I now invite Dr. Bobby Jones G, Professor of Pharmaceutics, St. Joseph's College of Pharmacy, Cherthala, to propose vote of thanks. Over to Bobby, sir.
Hope I am audible, Dr. Julie.
Yes, sir.
Okay. Respected President, distinguished Chief Guest, Guest of Honor, esteemed speakers, dignitaries, faculty members, students and participants. A very pleasant afternoon to all. It's my privilege to propose the vote of thanks on behalf of IPA Kerala State Branch on this remarkable occasion of National Science Day 2026, commemorating the discovery of the Raman Effect by the great scientist Sir C.V. Raman. Today's webinar has truly reflected the spirit of scientific curiosity, innovation, and interdisciplinary collaboration. At the outset, I express our sincere gratitude to Dr. P. Jayashekhar, President, IPA Kerala State Branch, for his inspiring presidential address and for his visionary leadership of the Indian Pharmaceutical Association. Thank you very much, sir. Also, my heartfelt thanks are also extended to Dr. John Joseph, Honorary Secretary of IPA Kerala State Branch. We are deeply honored by the presence of Professor Chandrabas Narayana, Professor at Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore. Sir, your enlightening inauguration and keynote address on Raman spectroscopy, basics used in drug discovery, has enriched us with valuable scientific insights and broadened our understanding of translational research. Thank you very much, sir. Our heartfelt thanks to the guest of honor, Dr. Sriraj Gopi, Chairman and Managing Director, Molecules Group, Thrissur, for sharing his distinguished perspectives and motivating the scientific community, especially women, towards innovation and industrial collaboration. Thank you, sir. We sincerely appreciate the National Science Day message delivered by Dr. Sabita M., Principal, Amrita School of Pharmacy, Kochi, which highlighted the theme Women in Science Catalyzing Viksit Bharat, inspiring us toward inclusive scientific advancement. Thank you, madam. I extend my heartfelt gratitude to the invited young scientist, Dr. Ashwati R. Devan, DST Inspire Fellow, Amrita School of Pharmacy, Kochi, and Dr. Dilin PM, Associate Professor, Dr. Moopan's College of Pharmacy, Wayanad, for their excellent presentations and for showcasing the vibrancy of contemporary pharmaceutical research. Your scholarly contributions are truly commendable. Special thanks to Dr. Anjana John, Principal, JDT Islam College of Pharmacy, Kozhikode, for the warm welcome address. Dr. Arun Rashid, Vice Principal, Al Shifa College of Pharmacy, Perindalmanna, and Dr. David Paul, Assistant Professor, NIPER, Kolkata, for introducing the dignitaries. And Dr. Julie Kurian, Professor of Pharmaceutics, Lisi College of Pharmacy, Ernakulam, for anchoring and guiding the session with grace and professionalism. I also acknowledge the dedicated efforts of the Organizing Committee of the Education Forum, IPA Kerala State Branch, Dr. K. Krishnakumar, Principal, St. James College of Pharmaceutical Sciences, Chalakudi, the technical support team, Mr. Prashant Kumar, Associate Professor, Maulana College of Pharmacy, Perindalmanna, and all the office bearers of IPA whose meticulous planning ensured the seamless conduct of this webinar. Finally, I extend my sincere appreciation and thanks to all dignitaries who joined us through Zoom as well as YouTube, making this program meaningful and interactive. Let us continue to uphold the spirit of scientific inquiry and collaborative progress. Thank you all. Good day.
Thank you, sir, for your kind words of gratitude. As we conclude National Science Day 2026, we carry forward the knowledge, inspiration, and purpose shared today. Today's deliberations have reaffirmed the power of science to question, to innovate, and to transform society. The theme, Women in Science, Catalyzing Viksit Bharat, has reminded us that inclusive progress is the foundation of a developed nation. May the insights shared today inspire our students and young researchers to pursue science with curiosity, confidence, and commitment, knowing that excellence in science knows no boundaries of gender. On behalf of Indian Pharmaceutical Association Kerala State Branch, I extend my sincere gratitude to our distinguished dignitaries, speakers, resource persons, and all participants, Bobisa, for making today's program a grand success. I also thank everyone who joined us through Zoom and YouTube and contributed to the vibrant spirit of this national celebration. I would also like to place on record my sincere gratitude to Dr. Ginu Isaac, Principal, Lisi College of Pharmacy, and Dr. John Joseph, Emeritus Professor, Lisi College of Pharmacy and Honorary Secretary of Indian Pharmaceutical Association, Kerala State Branch, and Dr. P. Jayashekhar, President, Indian Pharmaceutical Association, Kerala State Branch, for the constant support and for graciously extending the necessary facilities to successfully conduct and anchor this program. With this, we formally bring the proceedings of National Science Day 2026 to a close. With due respect to our nation, now we shall conclude today's program with the national anthem. I request all the participants, dignitaries to stand for the national anthem.